Okay, thank you, Wilsa, for this introduction. So yeah, today I'm going to show you what are exactly time varying ecological interactions, the why, how, and what, because I didn't know how to title this this presentation. Basically, it's the work that I have been doing here during my postdoc. But first of all, since I have some time, I would really like to just set the foundation of what we are talking about. So I will take my time to explain what are ecological interactions, then what is this time variability that we can add to ecological interactions, and then present you some results of my work at Doñana. So let's start by the basics. What are nodes and what are links? And it might seem too basic, but actually in ecology, I really like to, just at the beginning, define our actors, because sometimes I have been discussing for some minutes with someone, and then we realize that we were actually meaning different things, and that's not good. So nodes usually are species, but they can also be resources like light or water. But sometimes we also model individual-based networks. So nodes are individuals of several species, and nodes can also be locations, traits, or species characteristics, a lot of things. But usually just keep in mind that during the rest of the talk, we will see nodes as species. And then comes the tricky part, the links. So links usually are interactions, but interactions per se, it's also a very tricky concept in the sense that can be defined in very different ways and also can be measured in very different ways in the field. The most basic understanding are co-occurrent interactions. So basically present absence, and in that case of one particular species of or individual. So in that case, our adjacency matrix will be filled with ones and zeros. Then we can go one step further and add some weights, signs in these interactions and have and describe biotic interactions, for example, predation. And also other things that are not exactly biotic, for example, visitations, the frequency to which one pollinator visits a flower. And for example, in predation, you can have both definitions, a mechanistic definition or an effect definition, in the sense that predation can be an interaction in which one species eats another, that's a mechanism, eating, and also can be seen as an interaction in which the effect of population growth rate of the population of one species and the other is a plus sign. So for one species, it increases, and it's a minus for the other. For the other, the one that is eating, the prey, that abundance decreases. And that would be the effect definition. And actually, I really like this effect definition because it allows us to add dynamics to the network. So the most basic model to describe the dynamics of ecological communities is the Lotka-Volterra model. And there we have these two very important parts in complex networks. That is the structure of the network plus the dynamics that are embedded on the network. And in this case, the node variable that we are interested in is the abundance of a species, this Ni. That is going to be proportional to the intrinsic growth of a species, the R. That would be the growth that the species would have in isolation, let's say. Then plus interactions that are just proportional to the abundances or densities of the other species. So basically we have the interactions encoded in this matrix A, that is the agency matrix. And with this formulation, we could say that an interaction, so our links, symbolize the direct effect of a species J abundance on species I per capita growth rate, if all other species held constant. So it's like the partial derivative of these ODEs. And once this is set, we can actually say a lot about how the structure of this matrix A and the dynamics are unfolding. During the last decades, like 50 years or so, we have got quite a lot of results about how the structure of ecological interactions modified or affects the dynamics. And by dynamics, I mean the coexistence of the species, the stability of the community. So we have like recurrent patterns. For example, here I have several samples. Maybe one of the most well-known is this one in the middle. That is that usually networks that describes pollination have a nested structure, in the sense that we will have some species like this yellow butterfly that is a generalist that is connected with all the flowers, and then we'll have other species that are just connected to a subset of these flowers until we reach the orange butterfly that would be an specialist that is only specialized in this red flower. We also have the stress gradient hypothesis that basically coupled the interactions with the environment in which the species are living, in the sense that when the environment is very harsh, it has been found a predominance of facilitation of positive interactions, which might be a little bit counterintuitive. But when the environment is less harsh, less harmful, species actually are able to grow more. And since they grow more, they can compete more. So competition in the situation of no harsh environment increases quite a lot. But these are some examples of structure affecting dynamics. And then depending on how we model these interactions, we can also ask different questions, in the sense that we can also model space. For example, here is an early work that I did with individual-based networks, adding space. Or something that I have been doing here is modeling different type of interactions in the same networks. So in this example of Sonia Kefi et al., you not only have a food web, you also have competition, you also have mutualism. And when you consider the structure of all these compendium of links, you would get new patterns that emerge. Another way to visualize and study ecological networks is via multi-layer networks, in the sense that some locations can be modeled, or seasons can be modeled as a layer, and you can connect species through layers or within the layer, which is also very powerful representation. And then finally, of course, nowadays, I had to add the higher order representation of ecological interactions that has some benefits, let's say, but I'm a little bit suspicious about it, like too much buzzword, but whatever. And now what I want you to present you is what is exactly a time varying ecological network or ecological interactions. So the network that I have been showing you actually are time aggregated networks, in the sense that they symbolize a community that has been sampled through several years, and then you have gathered all this information together to create this network. So the matrix A is considered a species, a community property, and is fixed over time. When you simulate these load-coupled Terra functions that I have shown you earlier, the A is going to be fixed through all the simulation. But the thing is, can actually this network vary through time? And the answer is yes, definitely yes. They can change for a lot of particular reasons. One of them is the phenology, that I'm going to explain you now what it is. They can vary because basically there are migration and extinction of species. So if one node disappears, obviously the links, its links are going to disappear as well. Or they can vary because of environmental variability. The interactions that some plants are going to have with its predator when it's rainy or when it's sunny, it's going to be different. Here I have an example of phenology. That basically phenology is a study of the times in which in which organisms, in this case plants and pollinators, are simultaneously co-occurring through time. So in that example, if we just see the time aggregated network, we will think that the flower species 1 and 2, since they share one adult insect pollinator, they might be competing for the attention of this pollinator. But actually, we can see that they are flowering at different subseasons. So they are not going to occur in time. So there is no competition for that pollinator. At the beginning in season I, they will be one, the pollinator 3 will feed on flower 1, and then in season 2 on flower 2, and that's all. Then here we see that this time aggregated network, actually, if we know that there are some environmental variation in time or in space or both that affect species differently because they have different body size, different lifespan, functional traits, then you actually are going to have one particular representation of this network. It's not going to be disaggregation, but it can have different, in that case, the nodes are the same, but the links vary in a strength or basically in present or absence. And that give us a hint on how would network vary. So the most trivial one is by node turnover. So nodes appear and disappear, but also the links can change in every way we can imagine. They can change direction, the strength, or the sign. For example, here we have two seminal examples of time varying interactions that actually you can see the dates. They are quite new, like less, well, now not that new, but there is still a very an ongoing work in it. So, for example, in the paper on the left, like Caradona ecology letters, there is interaction rewiring in the sense that some interactions change the nodes they are appearing, and then of course the species turnover. That when one species, this pollinator disappeared, it also all its links disappear too. And by other other methods of inferring interactions, we can have here its color. It's actually one element of our interaction matrix, and we see instantaneous changes. So so in that paper, what they found is that there was a dominance of weak interactions and higher species diversity during some periods, especially summer, that are associated with higher stability and smaller population fluctuations. So we can now, with this framework of time variation, answer and think questions that we couldn't with a time aggregated network. And then from a network science perspective, the most straightforward way to represent these time varying interactions is a multi-layer network. So for each time step, we can have one different realization of a network that can, and the networks can be very different through time. And once we have that, we can relate, as the last example of the colored lines did, we can relate each realization of the network with one particular state of the environment or of the year, the phenology, and finally see how the interplay between my network structure and the environmental variability that is going on, how all these affect the stability and functioning on community, how the abundances fluctuate, a lot of things. So basically the big elephant in the room is like, okay, Violeta, you have been talking for like a lot of time about how good are these time varying networks. Why there are not more widespread? Well, because inferring networks is very difficult because you need long-term data that adapts to this framework. And it's even difficult to infer one network, one aggregated network. So let alone now 7 networks, for example. And so we need special rich data and also theoretical tools and metrics to now characterize and study these interactions, which is precisely what I have been doing. So in this case, we fly to Spain, southern Spain. And for the last 10 years now, there have been an amazing group of people that has been sampling every year several plots and regions. And finally, we have obtained almost 30,000 data points that are, so these data sets are rich enough to be able to study this temporal variation of ecological interactions. In particular, we have two types of data sets, one of wild solitary bees, and the other one is of annual plants. And please remember, they are not related. They share the environmental variable that is like its driver, but they are not. But the bees are not pollinating these plants. Are pollinating another plants, but not these. They are different data sets. Okay. And as I have just said, they do share an environmental driver that in this case is annual rainfall. And we are going to use this to add this temporal variability in the interactions. And I will say you how. Now, equations. Here, it's a discrete variation of the Lotka-Volterra model that I have just presented to you earlier. It's called Ricker model, but for us, what is important is that it has no temporal variation, neither in the intrinsic growth or in the interactions. So if you want to fit your data to this model, basically you would have all your temporal data or your spatial data or whatever, and infer just one single aggregated network of interactions A, and the intrinsic growth rates R. But we do not want that. We want to have time variation in our model. And how are we going to do that? Well, the way is very simple, too simple, because it's just a linear response. So now we have added a deterministic effect here that depends on the environmental variable each year. So basically, here we are assuming that the intrinsic growth rate now is a baseline plus an effect that is going to produce the rainfall. And since the rainfall varies through time, is there the parameter in which that is going to vary through time. And the same is going to be done for the interactions. We will have a baseline of interactions that is AIA plus one plus the effect of the environmental driver, the rainfall, that is this DAJ, and then some random effects because we are sampling. So here, so we have actually now two important matrices, the matrix A and the matrix B, that is this temporal effect that then is going to be multiplied each year by the rainfall that has fallen that year. And the combination of these three factors will create just one single network for each time step, and also growth rate, of course. So now we are going to have a snapshot, a series of networks, one for each year that these communities has been sampled. And with them, then we can ask the consequences that each structure has on diversity, on coexistence, on a lot of things. So how have we overcome the challenge that I told you? So for the long-term data, we do actually have it. We have done a little trick, that is that we have just focused on species that are annual, in the sense that one generation lasts only 1 year. So it's a complete turnover of individuals. And in that case, and in that case, 10 years is enough time to see some environmental stress going on. Then regarding the network inference, this is an ongoing work that I will be carrying on the rest of the course. Because for now we have used a generalized linear mix model, which is not the best, but it performs pretty good. But with data sets that are more sparse, it might not be the best. So I'm exploring more options like Bayesians and other maximum likelihood, but like hyper vitaminized. and now I'm going to present you the theoretical tools that we have developed to characterize and then to predict new things about these networks. So here you have them. These are the interaction networks for the data set of the plants, the bees. And of course, here you actually cannot see a lot of things. Well, you can see a lot of things, but you cannot quantitatively say anything. You can see that there is no node turnover, but the links are suffering every single thing that can be happening to them. There is link change in direction, sign, and strength. So we have came up with three important measures to ecologically meaningful measurements to characterize this time variation. One, it's called the temporality. It's from this paper of my colleagues, and basically it answered the question on how different are networks from time to time. So it compares two consecutive snapshots of networks to what would have been expected by random. And basically it answers the question of, but are actually our interactions network different? And yes, they are pretty different. So here we have our result of temporality versus the rainfall change between 1 year and the next for the whole network. And then in the small panels, we have only the network with positive interactions and the network of negative links. And the colors and dots represent the two different data sets. So even though this might seem a pretty obvious result, that with more rainfall, the networks are more different year to year, it is not, because when we are fitting a model, what would we could have faced the thing that the elements in the matrix A may be super big, and the elements in this matrix B could be very small. So at the end of the day, the effect of the environment would be negligible, but it's not. So this is a good starting point. Let's see, like, okay, so environment, do something to the networks. And the next thing that we measure that is very important for our ecological community is to look at the intra-specific interactions. That is a very fancy name to say self loops that, by the way, are not depicted here in the networks, but they exist. So basically the AII elements. And why we have measured them? Because in ecology, they are very important because they are regulation factor that creates more stability. So we have measured the intraspecific dominance, that basically if it's very high, that means that species self-regulate more, which creates more stability in the network. And here we see that if we plot this dominance against the rainfall anomaly, when that is our driver, when the values are very harsh, this self-regulating effect decreases a lot. So so now here the environment basically is creating a mechanism in which a lot of drought, let's say, destabilize the community in terms of the intraspecific interactions. But a part of the diagonal, we can also look at the interspecific interactions, that are the interactions that are among species and not the species itself. And here, this is the data set of the bees. We have found a very interesting thing, and is that if we just plot the facilitation, the positive interactions against the rainfall anomaly, we see that they increase more with negative rainfall anomalies like drought. And the opposite is found for the negative interactions, for competition. And actually, this thing of when we have droughts, that is a harsh environment, we actually found an increase in facilitation, and the other way around is completely coherent with the strength gradient hypothesis. So with these methods, we have been able to find an empirical validation of this hypothesis, not only in plants, that plants were the organism in which it was initially proposed, but also in other taxa like bees. Then here we have a little results about the intrinsic growth rates, because I know they are not networks, but they are very interesting, because since we have also made them vary through time, we've found actually that they not only depend on the rainfall anomaly, but also depending on the sampling site that the species is, they can vary enormously. And that's pretty cool, because that tells us that there are a lot of stochasticity and spatial contingency in the growth that a species can have depending on the site, which creates winners and losers of this environmental variability. There are some species that, for example, this Plantago coronopus, it's a plant. Depending on the site, it might have a positive growth or a negative growth with with rainfall, just depending on the site. So so far we have just characterized these interactions, and we have found that interactions do vary through time, and they do it in strength and size. We can also have this variation in intrinsic growth rates that are due to the stochasticity or the sites and the deterministic effects, that is the environment. And then we have that the networks vary with patterns that are coherent with theoretical prediction or expectations, like what's going on with the diagonal dominance or the rewiring. Well, that's a difficult word. Rewiring of our network according to the straight gradient hypothesis. And so good so far. I will is I think I have time to go a little beyond this characterization of the network. So far it has been like a descriptive analysis of what we have encountered in our study, but we can do more. What we can actually do? Well, we have a model, so we can actually do some simulations, because we have the R's, we have the A's, we have the B's, but we can try different rainfall profiles. So far, I have been using the rainfall that has actually fallen during the years of the sampling, but I can explore different scenarios to predict what's going to happen with my community, a community that I know how it's going to respond to this variation. And in particular, we set 4 like experiments. One was a constant value of the mean rainfall, and the other 3, the rainfall varies through time. In one case, it increases, the other decreases till a drought period, and in the last one, that is the most interesting, what is doing the rainfall actually is fluctuating, like in the real empirical case. And the results of diversity that has been measured by the Shannon index, that basically the highest better, in the sense that a higher Shannon index means more diverse species and distributed in a more homogeneous abundance profile. And we see that for the bees, our simulations, yes, only in the case of the increasing rainfall, our synthetic communities survive. But in the plants, it's very interesting, because the highest diversity index at the end of the day was obtained with this fluctuating rainfall scenario, not even by increasing rainfall. So it's a point in which increasing rainfall is not good for our communities. And then I don't know if I have time to present this, but if not, I can.
I mean, you definitely have like 4 or 5 more minutes, but if you need to like go over that, we could probably like go until 8:50 and then ask questions, get questions if you want. Yeah.
Okay, so I will just use these 4 or 5 minutes to draft one concept that we have implemented with time varying networks, and is this of structural stability. So basically, if we think about what ecological stability is, we usually think of a system that is unperturbed and still in its state. But actually, that is not what we see in nature. So we have come up with this structural stability approach, in which, in a set of taking instead of measuring the robustness to a small perturbation of a state, we characterize the robustness of our whole system to just have whatever the dynamical state, just having positive abundances. And basically what we do is to see how much we can vary parameters, the R's and the I, the interactions and the intrinsic growth rates, until we lose one species, basically. And funny enough, I will go. We can set several measurements for for this robustness, and the final one, that is the stability, does not vary a lot with the rainfall anomaly. That is actually very a result consistent with our simulation of a fluctuating rainfall. So there are some opportunities for a species to coexist, but then a species needs to be in the right place to profit these coexistence opportunities. So the conclusion for this part is that the results for these two taxa that are independent and not evolutionarily related is that the rewiring of the network actually behaves pretty similar. And then that since we have parameterized a model, we can also play with it and infer and predict what's going to happen for our communities with different time varying interactions. And that would be all. Oscar Godoy and Ignacio Bartomeus has been the two researchers I have been collaborating here for this project. And then also I did last year a very funny thing that I highly recommend everybody to try. That is complexity 72 hours. So basically I tutored a project with this fantastic group of students. And in 72 hours, we had to have some results presented as an archive report. So it was like a hackathon, but for complexity science. And I mean, if I were a PhD student, I would jump and sign in. And also as a tutor, it was a very delightful experience that I recommend to future postdocs and other researchers. And yeah, that's been all. Thank you very much.
Thanks so much, Violeta. This was amazing. I'll just jump in to kind of like promote complexity 72 hours from the perspective of a participant. I was there like
Ah,
2 years ago, and now I think they do it regularly in Spain. So yeah, it was really amazing. Like, I mean, yes, at points it was stressful, I guess, because we were just like rushing to write a paper in like 3 days. But it was really fun as well. And I mean, yeah, you get to like meet a lot of cool people and like talk about really good, like cool stuff. So yeah, definitely highly recommend. But yeah, thanks so much for this talk. Um, and we'll like take I mean, people, you can like either type your questions to chat or raise your hand and I'll just let Violeta know. In the meantime, I think, yeah, while you were like talking about all of this, what I've been thinking about is, okay, but like, how do we actually know like at what like level of granularity we need the data for, you know?
mm,
Because instead of like, okay, taking a data that is already available out there and like kind of like doing the analysis like from the other way around. But if I wanted to like study the interactions between some species, like how do I know like how like large of a, or like how long of a period I gotta um, just like watch them to begin with, but also like is it gonna be like an annual aggregation or like how do I know like it feels like it requires a lot of domain knowledge to be able to use like temporal networks and like to know like, or
Exactly.
either know the mechanisms that drive some sort of change, you know? Yeah, that's like what I was thinking. Okay, how do people know that much about like these environments to begin with so that like they can actually use these, you know?
Yeah, it's incredible actually the amount of detail that ecologists know about certain species. So yeah, as you were guessing, you really need to know what is the study community. And in our case, we were lucky because our organisms were all annual, which is very
Mm.
good because in that case, that generation time defines exactly your dynamics. Your the dynamics depending on our questions because our questions were regarding diversity, the relation of diversity to environmental variation, that of course you need to have several generations to actually see the effects. But if our questions were more like the amount of competition or resource partitioning that these communities have, for example, then actually just measuring weekly would be good in the sense that you would have these, well, it's actually this, what it is? Yeah, here. Here, for example, since your question is about when a flower and a pollinator are simultaneously um, coinciding in time, then your temporal scale needs to be way smaller, needs to be like days or 1 week, because flowers, at least in Doñana, are in bloom like several weeks. So you really have to be there when the interaction is happening. So yeah, it's highly system dependent, this temporal aggregation, and also question dependent. Hmm.
Thank you. Yeah. Okay, do we have any questions from the
I have a question. Uh, so first of all, Violeta, thank you so much for the wonderful talk. I feel like I learned so much, and there are so many papers now that I want to go back and read, even though I don't even do ecological networks. You made it seem so interesting. Um, I'm curious about like the consequences of this kind of work or like the implications for how we think about resilience in terms of climate change. So I thought it was super fascinating that you found the greatest diversity when there was more fluctuating rainfall, when there was more temporal variability. And so does that change how like the ecologists or other people that you work with or even how you think about how we can promote more biodiversity or biological resilience in face of climate change and global warming?
Yeah, definitely. For me, the most striking thing is that sometimes the architecture of the network, um, in principle, was able to sustain more diversity than the diversity that we were actually seeing. And actually, I don't know. In some sense, of course, this work is very theoretical, and it has been tuned to this system. And I would really, really like to see this philosophy being applied in other in other systems, in other communities that are more at risk. But yeah, but basically, I hope that adding time variation in the parameter facing climate change would be a thing in the next 10 years or something. Yes, but I mean, it's very, very time consuming to do this because, and baking up to the other question, you need to know your scale, your temporal scale. And I mean, sometimes for some systems, it's just, there is no time left. So you cannot do long-term study of it, because you have to implement your mitigation plan now and not wait several generations till you obtain these networks and understand how they change, because they might have disappeared altogether. It's so sad, but it's what it is.
Thank you so much.
All right, I'm just trying to see if we have any questions from the audience. Yeah, like you can either type it in chat or raise your hand, folks. Okay, we'll give it a couple more minutes, and if no questions, I'll take it as okay, no questions, I guess. But yeah, in the
Thank
meantime,
you.
I think
Yeah.
I was also, I mean, okay, that's like, I guess the kind of like the curse of social scientists, but like every time anybody talks about any kind of like system, I can't like help but like see the parallels in between like what they're talking about and humans, you know? So yeah, I was also like, especially when you like talked about some different like characteristics of like different or like structures of the networks. You like mentioned the difference between generalists and specialists, and I was thinking about I'm working on a project right now that where like I'm trying to see how that variation plays in a role, like plays a role in like how we do science, basically, like how scientists are
Oh,
generalists versus specialists. And I was like, oh wait, maybe I can yeah, like learn some stuff from ecology from that. But yeah, anyway, just wanted to mention because it's really interesting. I feel like every time I'm like listening to I don't know, a physicist, an ecologist, something, I'm like my brain directly goes into that direction of, oh, okay. But then I know like, yeah, social or like, I mean, I think it goes both ways. Like social scientists are not like the best in bringing like those different models like or approaches into their research, but also physicists are also like not super like well known about like how they approach those questions as well. So it's yeah, it goes both ways. And we were just intrigued to like try different things and see parallels, I guess, in general.
yeah, I'm always doing that. I mean, it's a curse, like, but in that sense, I remember during my PhD, I did some work on applying ecological concepts to social networks, like these pollination networks, like hashtags, where the flowers and the pollinators were the users. And then you also have one bipartite network, and you could also measure in-block nestedness, modularity, and those things. And so, yeah, I mean, it's not the first time it has been done. I really recommend you to dig into the ecological literature and see parallelisms, because I mean, that's also how science advances, like borrowing concepts or theories from one field and applying to the other. Yeah, it's going to be fun.
Yeah, definitely.
I'm happy to hear you say that because I was going to ask, like, how generalizable are these methods that you're using? Because I study brain networks, they're inherently time varying, and we think a lot about like methods used to study time varying brain networks. But I'm getting the sense that there's definitely space for these kinds of methods to be imported into other fields.
Yeah, definitely. I mean, the network characterization, I mean, they are networks, of course, can be applied. In fact, the first measured, the temporality measured, was actually measured first in baboons' social networks or something like that. So yeah, definitely. And then regarding the last part of the simulations and the structural stability, I mean, it will depend on the model, but I know there are some works with neural networks, but neural in the sense of brain networks. I don't know if those are your kind of system of study. the real neural networks or the computer neural networks, I don't know, but for the real ones, they have already been applied. So yeah, I think the philosophy is pretty straightforward. Of course, you will have to tune your questions, and then maybe the limitations are not around data, but around interpretability or something like that. But yeah.
All right. Yeah. Thanks so much, Violeta. It was amazing having you. I mean, I think it was like really a talk that is like kind of thought-provoking in different ways, at least for me.
Mm.
So yeah, it was like, thanks so much for joining us. And thanks everybody for being here as well. We'll have our next seminar in 2 weeks. So this year we're doing it bi-weekly again. So yeah, looking forward to seeing everybody
Thank
there,
you.
and much Violeta again. Yeah, bye.
Thank you for inviting me, guys. Take
For
care.
sure. Bye.
Thank you so
Bye.
much.
Bye.
Thank you. Bye bye.